Good morning. Quick announcement. If you go to B-Space under Exam 1 Resources, you're going to see a folder there that has several handouts on it, one of which is the exam handout. You are assigned to a particular room based upon your discussion section number. Not your not your exam or lab section, but your discussion section. So make sure you know that. Go to the correct room. And within each room, there's assigned seating. So there are detailed maps for each room on B-Space as well. Make sure you go to the correct room, because if you go to the wrong room, there will be no exam for you, most likely. And then you're going to have to scramble and come down to my office, and you're going to be running really late. So be on time, and make sure you know where you're going. Any questions about the exam? Okay.
Well, good morning, everyone. So today we're going to be talking about aerobic metabolism. And just to follow on Mike's announcement, you might have noticed that I sent out a message on B-Space to all of you yesterday. I've started to get quite a number of emails, typically long emails with a lot of questions. Fine if you have questions, but please don't do it over email. Okay, please come to office hours. You can come to my office hours. You can come to your GSI office hours. You can come to Mike's office hours. Lots of ways to get help, but not on email. Okay. So today we're going to continue our discussion. So Monday we started talking about glycolysis, and you recall that the introduction to all of this is the idea that cells have to get energy. They have to get energy typically from sugars and break them down into ultimately to carbon dioxide and water, and along the way harvest the energy of those various reactions that lead to the breakdown in a useful manner. And so that's really what we're continuing to talk about today when we talk about the citric acid cycle and ultimately oxidative phosphorylation. So basically, when we talk about oxidative, the citric acid cycle, we're going to be talking about a molecule of pyruvate, which you might remember from the discussion of glycolysis is the 3-carbon compound that results from the breakdown of glucose, right? And you remember that we get 2 molecules of pyruvate for every molecule of glucose that enters glycolysis. And once we end up with pyruvate, this compound can enter the mitochondrion and ultimately go into the citric acid cycle. And before it can actually get processed in the citric acid cycle, it has to be converted to a compound called acetyl-CoA. CoA stands for coenzyme A. It's one of the coenzymes that we looked at when we talked about cofactors and coenzymes, right? So one of the compounds that has an adenine ring at the center of it, right? It's a derivative of adenine. And this compound links the citric acid cycle to glycolysis. So the next slide shows the first step of this process. So this is before the citric acid cycle can actually get going. Pyruvate, and here it is, right here, has to go across the mitochondrial membrane, and once it gets across, it interacts with a large enzyme complex called pyruvate dehydrogenase, PDH, pyruvate dehydrogenase, and that enzyme complex catalyzes 3 steps of this reaction. Carbon dioxide is released. That's this portion of the pyruvate right here. We have a reduction of an NAD plus molecule to NADH, and we also have an addition reaction to coenzyme A. So coenzyme A is, if you look at the chemical structure in your book, it has a free thiol group, a free SH. And when it reacts with this acetate that's left over after the release of carbon dioxide from the pyruvate molecule, that acetate group gets added to the coenzyme A molecule through this thioester linkage right here. And that wavy line tells us that that's a very high energy bond. So this is effectively priming what's left of pyruvate, this acetate molecule, for entry into the citric acid cycle. And it's a critical step. It allows this molecule to be poised at the top of an energy hill and ready to go downhill in the subsequent reactions of the citric acid cycle. The other thing I want to point out here is that the pyruvate is a charged molecule. So this doesn't go freely across the mitochondrial membrane. And in fact, it probably wouldn't anyway, because initially the concentration of pyruvate is high out here, because this is where glycolysis is happening, right, in the cytosol. It has to be transported into the mitochondria and across this membrane by an active transport process using a transport protein. So that's what's shown right there. All right, so I wanted to mention one other thing about coenzyme A while we're on the last slide. So this is a very important step in sort of priming pyruvate to enter the citric acid cycle. And as we said, if we can't do this, then we can't make this acetyl-CoA molecule, then we really can't enter the citric acid cycle effectively with this molecule. And it turns out that coenzyme A is derived from a B vitamin, thiamine, and if there's in human populations that have thiamine deficiency, they develop a very bad neuronal defect that's a disease known as beriberi, B-E-R-I, beriberi. And it's really thought to be because they can't efficiently synthesize coenzyme A, and so that leads to a defect in this initial step prior to entry into the citric acid cycle. And it has a particularly severe effect on neuronal function for reasons that aren't not entirely clear, but probably have to do with the fact that the brain really needs glucose, right? It burns a lot of energy. All right, so once we make that molecule of acetyl-CoA, it's ready to go into the citric acid cycle. Now, in your book, you'll see that this is also called the Krebs cycle. And this is for a man named Hans Krebs, who really was the person who worked out many of the biochemical steps in this pathway back in the 1930s. So that's a very common way that you'll see this referred to, the Krebs cycle. And you'll also see it called the tricarboxylic acid cycle, TCA cycle. And you'll see why it's called that when we look at the chemistry that's actually going on in the reaction. Okay, so this is taking place inside the mitochondrial matrix. And the cycle is responsible for oxidizing organic fuel that comes in the form of pyruvate, which, as you now know, is now, as it goes into the cycle, is a molecule of acetyl-CoA. And in the process of each turn of the cycle, it's going to generate one ATP molecule, 3 NADH molecules, and 1 FADH2 molecule. This is another cofactor similar to NADH that can undergo reversible oxidation and reduction in the reaction. So this is kind of an overview of the citric acid cycle. So here's the starting material, pyruvate, going through this pyruvate dehydrogenase set of reactions to make acetyl-CoA, and then entering this cycle. And we'll see that it really is a cycle. It's going around and around, and at each turn of the crank, we're processing one molecule of acetyl-CoA and producing these products that are shown over here. So a couple of interesting things about this. So first of all, it was in the late 1940s that two scientists, Albert Leninger and Eugene Kennedy, showed that they could isolate mitochondria from cells, and that those isolated mitochondria were capable of catalyzing all of the reactions of the citric acid cycle. So that was really the first evidence that these enzymes are embedded within the mitochondrial matrix. And we can think about how this cycle is producing energy and how relevant it is to different kinds of organisms. In eukaryotes like us that are non-photosynthetic, the mitochondrion is the site of both the citric acid cycle and oxidative phosphorylation, as we'll see, where ATP is generated. and that's where most of the ATP in the cell is made, right? Remember from last time we said about 90% of the ATP is made as a result of oxidative phosphorylation. If we have photosynthetic eukaryotes, then what we find is that the mitochondria are the major site of ATP production in the dark at night, but chloroplasts are the primary source of ATP production in the light. We'll talk much more about that next week when we talk about photosynthesis. And then in bacteria, the actual enzymes that carry out this cycle are in the cytosol because they don't have mitochondria, right? And then as we'll see, the plasma membrane of bacteria contains the enzymes and complexes that carry out oxidative phosphorylation. All right, so the citric acid cycle has 8 steps to it, and each one is catalyzed by a specific enzyme, just like in glycolysis, right? So there's a very specialized enzyme or sometimes an enzyme complex that is responsible for taking each step, each substrate for each of these reactions and converting it into products. And these are highly specific enzymes, right? So we know that they have, just as we talked about when we discussed enzymes, they have active sites that are complementary to the substrates. They use the same principles that we talked about for catalysis to speed up the rates of these reactions. And interestingly, not every step of the citric acid cycle is a spontaneous or exergonic reaction. Some of the steps are actually endergonic, right? They are not spontaneous, meaning that equilibrium would lie towards substrate rather than product. And so we'll talk a little bit about that when we go through the steps of the cycle. And so the acetyl group of acetyl-CoA joins the cycle by combining with a molecule of oxaloacetate to form citrate. And then in the next 7 steps, we observe the decomposition of this citrate back to a molecule of oxaloacetate. So that's really what makes the process a cycle. We start with oxaloacetate, and we end up with oxaloacetate at the end. Importantly, the NADH and the FADH2 that's produced by the cycle are responsible for relaying electrons that are extracted from glucose or food to the electron transport chain. And that's what we want to really discuss today, is how we go through the citric acid cycle, we end up with these molecules of NADH and FADH2, and those then are responsible for driving the synthesis of ATP through oxidative phosphorylation. So let's go through these steps. Again, I don't want you or expect you to memorize the chemistry, the chemical formulas of each compound in the cycle, but I do want you to know all of the principles that we're discussing. The fact that the cycle has 8 steps, the fact that it's a cycle, the role of coenzyme A in activating pyruvate so that it can get into the cycle in the first place, and the energetic yield that's coming out of each turn of the cycle. All right, so these are all figures taken out of your book, so you can go and look later and read in more detail about each step of the citric acid cycle. And I want to point out that this is sort of a nice diagram because it shows you in red the carbons of the acetyl group of acetyl-CoA that are entering the cycle, and then what's happening, where they're going at each step in the cycle until we can't follow them anymore. Okay, so again, so this is the first step where we have oxaloacetate reacting with acetyl-CoA to make citrate, and here's the tricarboxylic acid, right? So this is why it's called the TCA cycle sometimes, is that here are those 3 carboxylic acid groups. This is just citric acid, right? It's a fairly simple 3-carbon molecule, and this is actually a very favorable reaction that's catalyzed. Again, it's an enzyme catalyzed reaction. Citrate synthase is catalyzing that reaction. And once citrate is made, it then reacts to form a compound called isocitrate, right here, with the loss of a water and then addition of a water in the process. And this is catalyzed by an enzyme called aconitase, and is actually not a favorable reaction in the sense that that energetically we would favor substrates over products. So it's an endergonic reaction, but because this compound is consumed very rapidly in the next step of the citric acid cycle, it actually drives the equilibrium towards the formation of isocitrate. So even though it's not a favorable reaction, the equilibrium drives it forward. Okay, so then we have, in the next step, we have isocitrate reacting to form a molecule of alpha-ketoglutarate. And here's where we see the first extraction of energy in the form of the production of a reduced molecule of NADH. Right? So we have reduction going on here, oxidation of isocitrate, and in the process, carbon dioxide coming off. And this is catalyzed by an enzyme called isocitrate dehydrogenase that catalyzes that reaction right there. And then once we get to alpha-ketoglutarate, we have another step that is where energy is extracted, again in the form of a reduction of NAD plus to NADH and splitting off of a carbon dioxide as alpha-ketoglutarate goes to a molecule of succinyl-CoA. Okay, so again, here we're seeing an important role of coenzyme A in this reaction, where we have this another example of a thioester occurring between this molecule right here and the coenzyme. High energy bond that primes this for the next step of the reaction. Okay, so then we get to succinyl-CoA, and this can then react to form a molecule of succinic acid, or succinate, which is shown right here. And note that now this is a symmetrical molecule, right? So we can no longer trace the path of these two carbons that we've been following that entered the pathway here, because we can't distinguish the difference. And this sort of brings up an interesting point. So does anybody know, how do we actually know where the carbons are going in the steps of the citric acid cycle? How would that be figured out? Does anybody know how you could do that? To label them. I heard label them. Yes, exactly. So you can label them with different isotopes and then just trace where they go in each step of the reaction by isolating the products of each enzyme and looking at which carbons get labeled, receive the labeled atom. Okay, so that's the way that this pathway was really traced out, was using labeled substrates for these reactions. So in this step here, where succinyl-CoA is being converted to succinate, you can see there's a lot going on. So we have the coenzyme being released, the free coenzyme. It can then be recycled and used to form another molecule of succinyl-CoA. And then we also have a phosphorylation step that's occurring, where we see phosphorylation of a molecule of GDP. So this is guanosine diphosphate, right? It's a diphosphate form of this nucleotide. It's being phosphorylated to form a molecule of GTP. And this, sort of similar to ATP, this is also a high energy molecule, and its formation is coupled to the formation of an ATP molecule, starting with ADP. And this is actually an example of another example of a substrate level phosphorylation step that we talked about last time, right? Where we're actually the phosphorylation, the phosphate group, is being transferred from a substrate to the ATP. Okay, so substrate level phosphorylation. So there we're making one molecule of ATP in the process. So that's another energy producing step in the reaction. All right, and then we get to this oxidation reaction right here, where succinate is converted to a molecule of fumarate, And again, this energy is derived by the reduction of an FAD molecule to FADH2. So another step where energy is extracted as these substrates are sequentially oxidized. Right, and then this is a hydration reaction. Hydration because water is being added here in the reaction as fumarate is converted to malate. And bear in mind that I haven't been mentioning the names of all of these enzymes, but there is an enzyme catalyzing each of these steps, right? They would not occur in a reasonable time frame on their own. They have to be enzyme catalyzed. So there's another specific enzyme catalyzing this reaction right here to form malate. And then finally, in the last step of the cycle, we have a molecule of malate being converted back to oxaloacetate. So we've completed the cycle at that point. And this is now ready to go back into the cycle as another molecule of acetyl-CoA comes in. And notice that, again, in this last step, we're extracting energy in the form of an NAD+ molecule being reduced to NADH in the process. All right, and so this is, again, sort of an interesting reaction because it's not at all spontaneous. In fact, the free energy change in the, going from malate to oxaloacetate is quite positive. It's about 29 kcals per mole. But the product, oxaloacetate, is consumed very rapidly in the next cycle of the citric acid cycle, so going from oxaloacetate over to citrate. So again, this is an example where even though at equilibrium we would favor substrate over product, the product is consumed so quickly that this sort of helps drive the equilibrium of this reaction towards the formation of oxaloacetate. And of course, we have an enzyme that's speeding up the rate of conversion anyway in that process. Okay, so that's the cycle. I want you to spend some time again with this, just as you're doing with the glycolysis pathway. In your book, they have a very nice sort of concise discussion of each step of the citric acid cycle that tells you a little bit about what's happening at each step. And you don't have to worry about the chemistry, but definitely you want to know the principles that are going on here. Yes? What do I mean by extracting energy? So basically what we're doing is we're building up a supply of reduced NADH and FADH2 that are going to be used to drive ATP synthesis. All right? So, and you'll see exactly how that works when we look at oxidative phosphorylation. And another question here. That's right. Where the energy comes from to do this? To NADH? Well, right. So again, we have to think about this in terms of the overall energetics of the cycle, right? So we're starting off with a molecule that's very high energy. You can sort of think about it like an energy diagram, right? So we start off with acetyl-CoA at the top of the hill energetically, and in each step of the cycle, it's dropping down, okay? And that's sort of a schematic way to think about it. Overall, that's what's happening. Individual steps, we might have some little bumps in the hill because some of the steps are not exergonic. But overall, we have to think about the overall energetics of the change in energy that's in going from acetyl-CoA to the final product of the reaction is a downhill reaction, energetically.
In that one step, what is the energy?
Well, it's a redox reaction, right? So again, even though this particular reaction is an endergonic reaction, we have an enzyme catalyzing this, and it's a redox reaction. So this reduction is part of the oxidation that's going on here. Okay, so this is an oxidation, this is a reduction, and even though the energetics at equilibrium would favor this, we're driving the equilibrium this way because of the consumption of the product in the cycle. Okay? Okay, so now we turn to sort of answer this question of, how is this really helping us in terms of deriving energy from a molecule of sugar? And to understand that, we have to talk about oxidative phosphorylation. And we're going to talk about a term called chemiosmosis. Chemiosmosis. And this is the idea that we can couple electron transport to ATP synthesis in a really fascinating set of reactions. All right, and so as we said, after glycolysis and the citric acid cycle, we have a lot of NADH and FADH2 that account for most of the energy that's extracted from food. Okay, but how does that work? How does that, what does that mean that we've extracted energy? And basically, it means that these are two electron carriers that can now donate electrons to what's called the electron transport chain. And it really is like a sort of a relay, right? It's a hand over hand where electrons are transported down this chain of complexes. These are mostly proteins, but not all, as we'll talk about. And ultimately, what this does is to provide the power for ATP synthesis via a process called oxidative phosphorylation. Okay, so we're going to talk about this pathway of electron transport. Again, where is this happening? Well, this is actually happening in the cristae of mitochondria. So remember that when we looked at the structure of the mitochondrion, you remember how the inner membrane of the mitochondrion had all those folds to it? You could see in beautiful micrographs that show this, where you have these inner folds of the mitochondrion that are called cristae. And the purpose for that is that it creates a large surface area where there's a lot of space for oxidative phosphorylation to go on. So thousands of these electron transport complexes can exist in that space because there's a lot of surface area for them. And most of the chain's components are proteins, but not all, and most of them are actually multi-protein complexes, so very large sets of proteins that come together to carry out this hand-over-hand transfer of electrons. And what happens is that these carriers are going to alternate between a reduced and an oxidized form as they accept and then donate electrons as they move down this chain. And again, the idea is that they're moving from what starts off as a high energy state to a lower energy state as they go through this electron transport chain, right, hand over hand. Right, so they drop in free energy as they go down the chain, and ultimately they're passed off to oxygen, forming water. And so we talked about oxygen being very electronegative, right? And it's basically at the bottom of the chain. It's the most electronegative component of the chain, if you will. So this is again a cartoon that's taken out of the book, and shows the complexes that are responsible for this electron transport chain. And they have it drawn here such that they have free energy plotted on the y-axis so that we can see that we start off at a relatively higher energy level than where we end up. So these are moving energetically downhill. So this is a downhill favorable process overall. And at the end, we reduce oxygen to water, but we don't want to do that at once. We want to do that in a controlled way. And so the way this happens is that these reduced molecules of NADH or FADH2 can enter the electron transport chain and bind to complexes that are sitting in this membrane in the mitochondrion, and then be passed along hand over hand to the different components of the chain until ultimately they are at the bottom of the chain and can lead to the production of water. So a couple of interesting things about this that I want to point out. First of all, these are complexes that typically have prosthetic groups bound to them. So you remember what prosthetic groups are, right? They're typically small organic molecules that bind very, very tightly to proteins and carry out an important function. And in the context of the electron transport chain, these prosthetic groups are really critical because they're actually typically what is doing the electron accepting and donating, right, is these prosthetic groups. So, for example, right here at the top of the chain, we have a molecule of FMN. This is flavin mononucleotide. And this is, again, derived from a B vitamin and is where electrons first enter the chain as a molecule of NADH is oxidized to NADH plus and gives up its electrons here. And then once entering the chain, these are passed along and they move ultimately into a number of other complexes. Interestingly, this molecule right here that's labeled Q is not a protein. This is a molecule called ubiquinone. And you'll also see it called coenzyme Q. So, if you go to the health food store and you can actually buy this as a supplement, coenzyme Q. And that's really what it is. It's a small molecule that's part of the electron transport chain. And it's sitting right here. And it's because it's a small molecule, it's not embedded within the membrane like these big complexes, these big protein complexes here. So it can actually move around in the membrane very rapidly. And that's part of the handoff mechanism, is to have ubiquinone moving in the membrane and helping to shuttle electrons down this chain through these large protein complexes. So again, these electron carriers here, they're alternating between reduced and oxidized forms as the electrons move along this chain. Also notice that FADH2 enters the electron transport chain at a lower, at a sort of a downhill step from where NADH enters the chain. So we get a little bit less energy out when we use FADH2 than when we use NADH, based on where it can enter the electron transport chain. And then these complexes here, these big complexes, you can see they have, you see these abbreviations CYTE, that stands for cytochrome. So these, the prosthetic groups in these big complexes are cytochromes. These are molecules that contain heme, which is similar to the heme that is in hemoglobin, contains an iron, except in this case the iron is not used to bind to oxygen like in hemoglobin, but it's used to bind electrons. And again, reversible oxidation reductions that occur as these electrons move downhill in this chain. All right, so electrons, this is sort of summarizing what we said. So electrons are transferred from NADH or FADH2 to the electron transport chain, and they pass through a number of proteins that include these cytochromes that have heme prosthetic groups, each with an iron atom that is able to reversibly accept and then donate electrons, and ultimately we end up reducing oxygen to water. That's what happens at the bottom of the chain. This is very important. Notice that the electron transport chain itself is not making any ATP. Okay, so we're not actually generating ATP in this process, and so you might be thinking, well, wait a second, you said that made ATP, and it does, but it does it in an indirect way that we're gonna talk about. Okay, but the chain itself doesn't generate any ATP. It's just harvesting electrons and ultimately reducing oxygen to water, and in the process oxidizing the NADH and FADH2 molecules that were made during the citric acid cycle or during glycolysis. Okay, so the chain's function is to break the large free energy drop from food to oxygen. Instead of occurring as an explosion, right, we want this to occur in a series of steps that are controlled so that we can actually use that energy that's released by breaking it into manageable chunks that can then be harvested. All right, and so to do that, we get to this very interesting process here called chemiosmosis. All right, and so this is basically a mechanism by which the electron transport chain, the energy derived from it, is coupled to the production of ATP. So electron transfer in this chain causes proteins to pump protons from inside the mitochondrial matrix out to the intermembrane space. So remember that the mitochondrion has those two membranes, right? An inner membrane and an outer membrane, and there's a space in between. And protons are being pumped from the interior to this intermembrane space. And that sets up a gradient where we have a lot more protons outside than inside. And so protons can then move back across the membrane. They can move down their concentration gradient, so that's a favorable reaction, right? And they pass through channels that occur in a complex called ATP synthase, of absolutely fascinating molecular motor. It's the smallest known molecular motor. It's really a little mill, and we'll see how it works. It's a really interesting molecule. And the ATP synthase uses this exergonic flow of protons to drive the phosphorylation of ATP. So now we see how that electron transport chain is useful for doing work in the cell, because it actually drives this phosphorylation step in ATP synthesis. And this is an example of what's called chemiosmosis, the use of energy in a proton gradient to drive cellular work. Okay, so Peter Mitchell first proposed this in 1961, and people thought he was crazy, right? Nobody believed it for a long time because it was so wild. But in the end, he was proven to be correct. And in 1978, he won the Nobel Prize for this discovery. Very important discovery in biology. All right, so let's look a little bit at how this actually works. So here is the ATP synthase that is sitting in the inner membrane of the mitochondrion. Here's the intermembrane space, so the outer membrane would be out here, and then here's the interior mitochondrial matrix. And so the electron transport chain would be sitting over here somewhere and producing this gradient of protons. So we have a higher concentration of protons in the intermembrane space. And then a really cool reaction happens. So these protons, there's now a concentration gradient. They want to get back inside the mitochondrion based on the just diffusion, right? They want to flow down their concentration gradient. And the way they do that is they interact with a part of the ATP synthase called the stator, just analogous to a sort of a real motor, right? And they enter this stator. From there, they bind to sites on what's called the rotor in the ATP synthase, and they cause conformational changes in these proteins. And by the way, remember that these are all proteins here, that cause the rotor to spin. It spins like a real rotor. It moves around in a circle, and each ion that binds here makes one turn on the rotor before it jumps off and enters this other little pore in the stator, and then is eventually shunted out into the mitochondrial matrix. And in the process of this rotor turns an internal rod that's connected to a set of catalytic residues down here on the ATP synthase. And these are this is the part of the ATP synthase complex that is responsible for actually synthesizing ATP from ADP and inorganic phosphate. And so as these protons enter and spin the rotor, that drives this catalytic cycle. A guy named John Walker in the UK was the person who solved the structure of ATP synthase and actually figured out a lot of the molecular details of how this works by looking at the structure. But a crystal structure is a static molecule, right? Or a static picture, a snapshot of a molecule. It can't really capture the kind of motion that we're talking about here. To do that, there was a very interesting experiment that was done that I'm going to show you. This is discussed in your book, and it's a really classic experiment and beautifully illustrated one of the aspects about this motor that's critical to the production of ATP. So the idea was that, so if we go back to the last slide for a second. So if you look here, you can see that protons enter this rotor, and the rotor spins in a specific direction, right? It spins in this direction here, a clockwise direction, and that drives the rotation of these catalytic knobs down here, which leads to ATP synthesis. So you might imagine that if the rotor spins clockwise and it makes ATP, what would happen if it spun in the other direction? ATP hydrolysis, right? So you might just run this reaction in reverse. And in fact, that was the hypothesis, was that if this rotor spins in this direction, the clockwise direction, ATP synthesis would occur, but if it were kind of artificially made to spin in the other direction, ATP hydrolysis would occur. And so there were several groups, several research groups, academic research groups, and also a company that was interested in really trying to figure out a way to test this idea directly. And they came up with a really cool experiment, and that's shown right here. So the idea was they could take a piece of the ATP synthase and they could attach it to a nickel plate. And this was done, so you can see here we have the catalytic knobs, and then here we have this internal rod. And so remember that in the ATP synthase molecule, this rod would normally be attached to that rotor, right, which would be driven by protons dropping onto it and spinning it in one direction. And so what they did was to replace the rotor with a big magnetic bead that's shown right here. And this is attached to the rod. And so the idea was that they could, because this is a magnetic bead, they could use a tiny magnet to artificially spin the rotor, spin the rod, in either the clockwise direction or the counterclockwise direction. And so that was the technical setup of the experiment, and then they had to have a way to measure whether ATP was being produced or not in the reaction. And the way that was done was to do this in a solution where they had a reporter protein that could hydrolyze ATP if there was any ATP around, and each time ATP was hydrolyzed, a photon was produced, so you could actually measure it, you could see it, detect it. And so that was the experiment. And so they did this reaction, and they could spin the bead either in the clockwise direction or the counterclockwise direction and measure whether ATP was being made or not by looking at the number of photons being produced by this ATP hydrolyzing reporter. And here's what they saw. So these are sequential trials, sets of experiments, where they either did no rotation, they rotated the rod in one direction or in the other direction. And then they counted the number of photons that they could detect in each case. And what you can see is that when they rotated in one direction, they saw a lot more photons produced consistently, these yellow bars, than they saw when they rotated in the other direction, in these blue bars, or when they did no rotation in the gray bars. So that really suggested that this idea was right, that the rotor spins in one direction and drives synthesis. If it goes in the other direction, the reaction goes in reverse. Very cool. I love that experiment. Okay, so the energy stored in a proton gradient across the membrane couples the redox reactions of electron transport to ATP synthesis through this really cool motor called ATP synthase. The proton gradient is called a proton motive force. You'll see this abbreviated PMF sometimes, proton motive force. This emphasizes that it has capacity to do work. The movement of protons is what is driving the chemical synthesis of ATP. All right, and so I want to show you this slide right here. Again, this is from the book that shows the travel of how electrons are basically moving through these complexes in the electron transport chain and eventually driving ATP synthesis through this ATP synthase complex over here. The gold arrows are basically showing you the path by which electrons move through this electron transport chain. And again, remember that there are these three big complexes called complex 1, 2, and I guess 1, 3, and 4 they show you here. Okay, 1, 3, and 4. And these are multi-protein complexes that are sitting embedded within the membrane. So even though membranes are kind of fluid, right? We talked about the fluid mosaic model for membranes. So these can move around to some extent. They can't move that fast because they're pretty big complexes. And in contrast, we said that coenzyme Q, this ubiquinone molecule here, is a small molecule. It's hydrophobic. It's sitting in the interior of the membrane, and it can actually move around very fast because it's a small molecule. It helps in this shuttling process. And so does a molecule called cytochrome C, which is also a smaller molecule protein that can move around and help shuttle these electrons between these bigger, bulky complexes. And again, you can see that we have NADH or FADH2 entering the chain at two different points and leading to the transfer of electrons that can then be moved through this chain. And you see that at each handoff step, we have production of protons that are shunted out into the intermembrane space where they set up this gradient that is then used to drive ATP synthesis. So this just shows you that this part of the pathway is called the electron transport chain that we talked about. This part with ATP synthesis is the process of chemiosmosis, and the whole thing is oxidative phosphorylation. Okay, so the whole pathway is oxidative phosphorylation, the handoff of electrons, setting up this proton gradient that drives ATP synthesis over here. Okay, so finally, let's just do a little accounting of the energy that's coming from all of this. So on Monday, we started off with glucose, right? And we talked about its entry into the glycolytic pathway to produce ultimately to produce pyruvate that then goes into the citric acid cycle and drives electron transport through the production of NADH, right? So this is sort of the overall set of things that are happening: glucose getting broken down, NADH getting produced, driving the electron transport chain by dumping its electrons into this hand-over-hand pathway that builds up a concentration of protons outside the membrane, producing a proton motive force that drives ATP production. Now, let's do a little accounting. So if we said last time that for each molecule of glucose that is completely oxidized, so if we say glucose oxidation, overall delta G for that reaction is minus 686 kcals per mole, right? And, oops, sorry, I didn't write it on the other board. And when ADP plus an inorganic phosphate goes to ATP, this costs about 7.3 kcals per mole. And if you look in your book, they tell you that there are roughly something between 36 and 38 molecules of ATP that are made per molecule of glucose. So we can multiply this by, let's just say 38 ATPs per glucose. Okay, and we end up with a number that's about 277. That is roughly 40% of the overall possible energy that we could get from the oxidation of glucose. So it's pretty good, right? We're actually capturing 40% of the energy of the oxidation of glucose in this process. And by contrast, if you think, it doesn't sound that great, in our car, when you put gasoline in your car, you're getting somewhat less than 25% energetic efficiency, right? So actually our biology is pretty good at harvesting energy. And I want to tell you one more thing, because this came up in office hours, and I thought it was a really interesting question. So this is from your book. But the question was, what about in bacteria? So we've been talking about in mitochondria, but what about electron transport in bacteria? So remember that in bacteria, so we keep saying that these enzymes are for oxidative phosphorylation are embedded in the inner membrane. And in bacteria, remember that they also have a structure where they have an outer membrane. This is for a gram-negative bacterial cell wall. If we have a gram-positive bacteria, then that also has even an extra layer of lipopolysaccharide outside of the outer membrane. But again, you have this interior space where protons can be pumped, setting up a gradient that drives ATP synthesis. And in fact, very interestingly, a lot of biologists think that mitochondria actually derived originally from gram-negative bacteria that invaded cells, and because they were very useful, they were effectively symbionts, they were able to make energy, and that is what led to the evolution of eukaryotic cells. Okay, so have a nice day. I'll see you guys on Friday.